Cool. Now I'm speaking to the whole world. Great. Yeah.
What is the output of the network? You give the position in space of each.
Yeah, it outputs the positions of each atom and each residue in the protein. So that's the output. Yeah. So let me first give like some sort of high-level overview of what AlphaFold is as a model. So one thing about it is it's quite a deep model. So it has 192 blocks, which is 84 parametric blocks, and then we run it like a recurrent network 4 times. But it's also quite a small network by today's standard. So it's less than 100 million parameters, whereas, for example, GPT-3 is 175 billion parameters, so 2,000 times larger. But it's also a network which takes quite a long time to run, again, by the standards of today's network. So it's like tens of seconds to hours of runtime, depending on the protein length. So if it's a very big protein, it will take hours. If it's a small one, maybe 10 seconds. And the data it was trained on is, so there is we have labeled data, which is roughly 170,000 of 3D structures, and they are taken from Protein Data Bank, which is an absolutely incredible resource. So all experimentalists in the world who are working on academic research will upload their structures to this database, and you can access it. It's machine readable, it's high quality, so it's an absolutely amazing resource, probably one of the reasons that AlphaFold was possible. But we also use some unlabeled data. So we have roughly 350,000 deduplicate sequences taken from UniProt. So these are the ones for which we don't know the structure. But what we do, we predict the structure and then use it, we distill it in the next iteration of models. So we use it as the ground truth for the next step. So that's basically the training data. I will point out that the training data is very small by today's standards again. Again, it's nothing close to training on whole of internet as we discussed yesterday. Yep.
Which organism did the data come from?
All sorts of organisms. There is a very strong bias, of course, to the ones which are studied most. Like for example, yeast and human, like are probably the, I'm not sure, but I suspect they're the 2 most common ones.
But it's not just humans. So you
No.
a version of a protein that's conserved in humans, and then you would have the same protein in yeast or in animals.
Yes, it's basically everything the structural biologists will study, right? So whatever they study, they will deposit. But there is, of course, a huge bias towards things which are easy to work with or which are interesting from the point of view of medicine. So, yep.
Why do you use the unlabeled data? I mean, if your talk is wrong and you're using predictions as a target, then you're kind of reinforcing errors.
That's true. We try to filter out the less confident predictions, so we have some uncertainty measures. So we try to only train on the ones which we're confident about. But I think that's true. But the problem is the labeled data set is very small. So adding some unlabeled data really helps with the training. But yes, it can cause some sort of loops where the biases are amplified. Cool.
The runtime is the runtime for the training of the network on the database, or the runtime on one protein to determine its structure?
It's a runtime for one protein to determine the structure. So it's probably one of the most expensive networks people routinely use.
How comes it's so long, given the fact that the number of parameters?
It has cubic runtime in the length of protein. So transformers are quadratic. This one is cubic, which is the main reason for that.
And the training time?
The train time, what is it? I think something like about 2 weeks. Yeah, so 128 devices, I think. So yeah, that is much more standard for modern networks. Probably also on the smaller side by now. Yeah. Cool. Let's move on. So this is the overview of the model. So we have 3 stages. The first one is the embedding stage, where we incorporate the evolutionary information. So this is the genetic database search I mentioned. You make multiple sequence alignment. You also make the residue pairs, which I will talk about on the next slide. And we have something called templates, which is basically similar enough proteins for which we know the structure, but I will not talk about that. So in the next step, we try to determine the relationship between the residue pairs. So this is like a very deep network, which iteratively tries to improve the representation for the residue pairs. And at the end, we have a block which directly outputs the structure called structure module. So this is a very complicated model. So I will only show you one example, but we have the paper, which includes 60 pages of supplementary materials describing everything in absolutely minute detail. So everything is fully described, including nice diagrams like this and algorithms. And we have also open sourced the code, and the community is already using that to build on our work. So if you want to know more details, you can check this out. But let's dive into one block in detail. So this is the EVA-former block, which is where the network spends most of its computation, probably something like 99%. So the two main concepts, the two main representations here are the multiple sequence alignment and the residue pairs. And just to be clear, this matrix is basically in each position you have a vector, right? A vector of activations, right? So you have a vector here, and vector here, here, and here, right? So all of those are actually 3D matrices. Now, the first update we do is attention on the rows of the multiple sequence alignment. So within each row, we will do a self-attention operation as described yesterday. But to make this more specific to what we know about the protein problem, we will augment this with the network's belief about residue pairs. In practice, this just means that you remember this attention probabilities map, right? So we will just add a bias which is projected from the residue pairs. So each residue pair can say, yes, I want this pair to interact more, or maybe I want to interact less. And that will allow the network to encode information. Yes, I know these things in context, so maybe they should talk more. Or maybe it's not certain, so they still want to interact. Yep.
But attention is with regard to what follow what. What we saw yesterday.
Yep.
What amino acid follow knowing a serial.
Yeah, so here it is so yesterday it was the left to right attention, so like autoregressive, what it's called, and this one is unconstrained. So everyone can talk to everyone in this case, because basically we don't want to generate these proteins, so it's fine to talk for everyone to talk to everyone. So on the next step, we need something to go from the multiple sequence alignment to the residues. So somehow inform the residue pairs about the multiple sequence alignment. And what we do is an outer product operation. So the way to think about this is you take the columns of this matrix and just compute correlation between them, between all pairs of the columns, right? And then you get number of residues by number of residues matrix. So that's basically the intuition behind this operation. So you just compute correlation. So what correlates to what? And then use this to update the residue pairs. Okay? Makes sense. So that allows you to incorporate things like coevolution, as I described, like generalized correlation. And finally, the bit which is probably the most interesting is the triangular attention. So this is a residue pair interaction inspired by geometric interactions. And let me try to go through this. And this will be probably the most dense slide of the whole presentation. So sorry about that, but hopefully some of you will find this interesting. So how does this work? Within the residue pair representation, the starting point is within each row. Again, all residue pairs will talk to each other. So for example, within this row, IGA will talk to IK and to all others, right? So everyone talks to everyone here. Now, you can also view this through the graph representation, basically as we described yesterday with the graph neural networks. So, but the difference is here, we will draw it as nodes will be the residues, IJ and K, and IJ will be an edge here, right? So this position corresponds to this edge, okay? So now if we think about this, these edges should encode some spatial relations between the residues, right? That's probably what they should represent. And let's say they are something like distances in 3D. Now, one of the most interesting properties of the 3D space is the fact that we have triangle inequality. And we also have transitivity. So we would like to represent that within our neural network. But if we think about this, this probably means that if we want to encode something about a triplet, like let's say we want to encode the fact that the distance of ij plus the distance of jk should be greater than the distance ik, right? You should have some interaction which encompasses all three of these edges. Whereas currently, we only have two. ij will talk to ik, right? So there is a missing third edge. And what we do here is we basically add this extra third edge. So if we write out the equation for the attention weights, you already have ij and ik. And what we do, we add a term which depends on jk. Okay, so we basically add this third edge to the triangle. And the way we implement this is just a bias term, which is projected from the same matrix. So basically, we just take the features here and use it to project some number to add here. Okay, make sense? Yeah.
There is a sort of subtlety there, right? Like in the fact that it's not the nodes that are talking to each other, right? But actually a sort of upper layer of abstraction where it's actually edges.
I think this is crucial, to be honest. Yes, it's probably even more crucial than the exact thing I am describing here. The fact that we have the pair representation seems absolutely crucial. Yes. So you have an explicit representation for pairs of residues. And that's not something that transformers typically do. And the reason for that is it's very slow. I think this mostly makes sense in cases where there are very complicated interactions between your tokens. And then you may want to do something like this.
So are we expecting that the interactions are symmetric for the pair representations?
Yeah, but here they shouldn't be necessarily symmetric. If you think about, for example, relative positions, they will not be symmetric, right? So we basically symmetrize before we want to project something that should be symmetric. Yeah.
I think there is something. How do we encode? We want to predict geometry.
Yep.
3D. How do we encode this geometry?
That will be in one of the next slides. Yeah. Yeah. Okay. Shall we move on? Yep. Okay. So that's basically how we encode the The first thing is it's end-to-end folding network. I think this was one of the first ones to actually go end-to-end instead of using something like gradient descent at the end to produce the structure. And there is pretty interesting idea here, which is the fact that the protein backbone, which, as we know, a chain, is encoded as a gas of 3D rigid bodies. So it's there is no inductive bias directly to encode the fact that it's a chain. Instead, what we do is we fit a rigid body, like a triangle, into each of these residues and just let them flow freely. So the network can put them anywhere. It is not actually forced to put them close by. And then we use an SO3 equivariant transformer, as Seb talked a little bit about equivariance yesterday, which updates these rigid bodies. So they basically, the network starts to move them around such that they start resembling a protein structure. And it happens very rapidly. So basically, you can see like at the first iteration, it's almost a protein structure, except some scales are wrong, but then it's quickly adjusts and refines. Okay, and
at the start of the iteration, you already know the local structures, like the LAs.
This representation is initialized just at what we call a black hole initialization, where everything is just put to the origin. So it's like all the residues are on top of each other. But the network starts moving them to the correct places very rapidly by using the features from here. So it knows these features which already encode a lot of knowledge about this protein and can start building the structure. Does this make sense?
I was just saying this because on the figures,
yeah,
it's changing, and on this one, and iteration 1, you already have this protein
yeah.
ACs.
So
you already know identify these typical structures?
No, you start from basically nothing here. So no, iteration 0 is essentially everything at the origin, like all the residues are the same, so you can't really visualize that, at least with the standard tools. So it's, yeah. And at iteration 1, it already looks a lot like a protein structure. So the network can do this in one step, essentially, is what this is saying. Yeah. Yep.
So if the structure of the protein is understood at iteration 1 already, and it uses this prior attention matrix,
Yep.
how do you then compute that attention matrix for protein you've never seen before? How do you run inference?
So you basically start from the multiple sequence alignment and you run a lot of iterations here and you get this pair representation. I think it's mostly the pair representation, which allows you to do that. So essentially it encodes the distances already, and then you can use that to build the structure quite easily. So it's not such a hard problem once you understand the distances.
But you have to have some sort of prior knowledge about the structure of the protein and its evolutionary history.
Yes.
So if I synthesize a brand new protein in the lab, there is no, I mean, I wouldn't expect AlphaFold to be able to fold it properly.
It does quite well on the designed proteins, what it's called, which are basically people designed with some, using some tools, because basically these tools also have a lot of prior knowledge built in them. Like the protein sequences are quite specific. It's not quite as well on what is called orphan proteins, which actually have no multiple sequence alignments. You need like a few dozen alignments to make this work.
And do you have an estimate for what the minimum is?
Yeah, if you, I would say maybe starting from maybe 20, you can hope for quite a good solution. If you have like 10 or so, it's like a bit of a gamble. Sometimes it works, sometimes it doesn't. Which is still much better than the previous methods, which seem to use like thousands. But still, yes, it does need some information to get this off the ground. Yep.
Which database you are using to align the input sequence?
So the alignment was that was done with the. So it uses UniProt, I think, Magnify, and a database called BFD.
And what if your sequence is novel and does not align? What will happen?
So you will have a poor MSA, I think, then. So multiple sequence alignment will be bad, and probably the predictions won't be very good.
In your database, you don't have all the proteins involved. So
Yeah.
when a researcher sequence, new
Yeah.
sequence, you will not align your system. So what will happen?
Yeah, you will just get unconfident prediction. I will get to the confidence measures in a few slides, but yes, that does happen, but it doesn't happen very often. Basically, most proteins do have quite a few alignments. Cool. So I described this quite complicated system, and we tried to run ablations to figure out what were the most important parts. And for better or for worse, the answer was like most of the pieces were quite important. Yes, the question?
You started all the residues in the same place.
Yep.
And it means that the order of the residues is not important.
It is important, but it's encoded again in this evoformer part. So evoformer part knows the order, and then the structure module can exploit that to build a chain. Yes, so it's okay. Yes, so this initialization at 0 is basically just similar to random initialization in some sense, right? So it's just uninformative. Yeah, yeah. Cool. So we tried quite a few ablations and tried removing different parts of the network. And for most of them, you get quite a small drop in performance. Although for some, it's getting quite a bit bigger. For example, if you, like 5 GDT points, if you remove this triangles idea that I described. But it seems like the most important thing was the methodology of building this protein intuition into the model. And another point is that if you start removing multiple components from the network, you get quite a dramatic drop, which also suggests that probably there is interdependence between different components. Cool. Now, yeah, that's invariant pointed attention. That's basically this thing, the equivariant transform architecture. Cool. So how does AlphaFold understand proteins? So just a few points here. One is that the computational structure prediction is usually underspecified. So I described this bright picture of going from sequence to the structure. The problem is it abstracts away so much of the world. So there are things like oligomeric states. So some proteins will only fold if there are a few other copies of the same protein around. So basically can't get a structure of just individual chain. It will always be a complex with other chains. Sometimes there are ligands like small molecules or ions like here. Some proteins bind DNA and will not be structured without DNA. You can change experimental conditions in the lab and you will get different structures. You have multiple conformations of proteins that are actually moving around. And the amazing thing is the network seems tolerant to this missing context. So here's one example. This is zinc binding. So zinc is a metal ion. And you can see that the network perfectly predicts the side chains which coordinate the zinc, despite not knowing that it's there. So it has predicted that there will be a zinc and then put the side chains exactly in the right place for this zinc. This is another example. So AlphaFold only predicts single chains, but it predicts perfectly the structure of a trimer. So if you basically just take a protein and put two more copies of it, they align perfectly with the experimental structure. So it clearly has a concept of multimeric proteins despite not being told that. Now, to interpret the model, one thing we tried was predicting the structure from intermediate layers of the network. So we just take the features from the intermediate layers and use the same structure module to predict the structure at that stage. And we freeze the weights while doing that, so the network, like, it's not backpropagated into the model. Yeah. 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 So essentially, you can try to get the evolution of the structure through the network by doing that and try to figure out what is it doing, how does it look like.
Third time, sorry.
No worries. Cool. So, and this is what the what it looks like. So this is one of the harder proteins, and you can see that it resembles search quite a bit, which is surprising because there is nothing about architecture which would suggest search necessarily, but it does look like it's sort of trying to arrange things such that the structure will make sense. And this is an example of a much bigger protein. And the interesting thing here is that the domains, like the small packed bits, show up quite early, but then it keeps refining their arrangement relative to each other until the very end. And then the question is, like, is it actually important? Does it actually keep improving throughout the network? And you see this. So the x-axis are the blocks of the network, and y-axis is the GDT. And you can see that it keeps improving for a while. And in this case, it basically keeps improving until the end. So you would assume that if you kept, if you added more iterations, it would be better. And that's what you sometimes find. So it does seem like there is some component of search in there where it keeps refining the structure for as long as it can. Any questions about this? Yeah.
Can I go back? Can I find a structure that isn't optimal and then go back and reverse it?
Reverse in what sense, sorry?
As in, you find, for example, two recipes that are very likely to interact, but then the rest of the protein doesn't align with that. And can it break that interaction?
Yeah, definitely. Yes, that it can definitely do. Yeah. Cool. So AlphaFold and the biology community. So after we have released the paper and the weights, people were very enthusiastic because it was actually amazing for me as a machine learning researcher because it seemed like people have started using that for their own research and that actually gave them new capabilities. Like here, someone was able to get the structure of a protein which wouldn't get expressed and purified. So they were able to continue on with their research. People also started using this in their own tools and hacking with the code base, trying to do things like, predict multimeric structures, whereas the network was only trained for single chains and things like that. And let me give you like a few lessons from this, like one of the interesting things. So maybe the most interesting thing for me as a machine learning researcher was that communicating confidence is key for this. So the typical machine learning model we will build in computer vision or a natural language will not communicate confidence at all. Like you get something, but it's usually like, it's wrong and you don't care, basically. So AlphaFold outputs two confidence metrics, PLDDT and PIE. So PLDDT basically tells you whether you can trust the local neighborhood of a residue. So like if it's blue, then probably everything around it is mostly correct. So like all of this blob, you can probably trust. Whereas if it's red, like here, you shouldn't trust that. And PIE gives you a pairwise measure of confidence. So should you trust the interaction between these two things? So dark green means yes, and light green means no. So for example, these two residues, you don't know how they interact. And it turned out to be extremely important for sharing these predictions responsibly. So people started using this in the paper. So if they're using a computational prediction, they will share this because this gives the people reading the paper a baseline of like, should you trust this prediction, which is incredibly important. But it also actually opened up some new use cases. So one of the most obvious one is domain segmentation. So domain is basically a block of protein which folds on its own, and you can often see them like this is probably a domain here. You can use it for software integration. So if your software wants to know like the level of confidence in something, like maybe because it wants to gauge flexibility of the proteins or something else, that is very important. Detecting interactions, so like the probability that in the PIE maps often correspond to interactions, but also disorder prediction. So if you have something like this, which looks like not a protein, oftentimes this is highly correlated with disorder. So proteins which don't have actual structure. And another thing people use this for is ranking models. So sometimes people will run thousands of models and just rank them by their confidence. And that seems to work as well. So there's this host of applications for having good confidence measures. And I think we should look into this more in other domains. Now, probably some of the biggest wins for AlphaFold has been on the interface with the experiment, which is again quite surprising because basically we're talking about this, like how you can sidestep the experiment, but in practice, the biggest wins have been at the interface with the experiment. So for example, the model may help the experimentalists to interpret the data they have, or even maybe even more importantly, it helps them to understand what experiment they should run. So like as an example on the right, here's a cryo-EM density. Like it's this sort of a blob of densities. It's not very precise. But if you use AlphaFold, you can fill in the atomic details. But then sometimes you will see that there is a disagreement, right? And that means you shouldn't trust the AlphaFold model in that part of the prediction, right? So there is this sort of complementarity, which is extremely important. And maybe one takeaway is that you shouldn't like over-index on doing everything you can in silico on your computer. Sometimes it's worth asking like, okay, we will build this model. How will this feed into experiments that people will want to run, right? Cool. And maybe my favorite example of the applications of AlphaFold so far was studying of the human nuclear pore on the left. So this is a humongous complex that is the gateway to the to the nucleus of the cell. So basically, it guards your DNA in your cells. And this is a huge thing. It's like I don't know, 100,000 residues, something huge like that. And the existing experimental methods can only give you basically these blobs of density. So you get like these blobs, but you don't get atomic detail. So what these researchers did was using AlphaFold to fill in all the atomistic details of the structures using AlphaFold. So basically, they can see that, yes, this blob, I can fit in the AlphaFold prediction there and get all the atomic resolution. And this way, they have built the entire atomic scale structure of this nuclear pore, which is super important for understanding how it works. And on the right is the intraflagellar transport trains. I don't think I can pronounce that, but basically this is also a similar application where you can use this AlphaFold predictions to understand experimental structures, experimental results, and build up your models. Cool. So another thing we did to benefit the biological community was releasing the AlphaFold protein structure database in partnership with EMBL EBI. So currently it includes over 200 million predictions. So that basically is all of UniProt. So essentially, for most of proteins you may be interested in, you can probably find AlphaFold prediction in there. And it includes the confidence measures and everything. So you can use that in your research. And then the question is, how can you use that? And I would like to highlight FoldSeek, which is an incredible tool which allows you to search for structures, even when against very large databases like the AlphaFold database. So if you have a structure, you can find everything that looks like it in this database. And one of the research application is, so in this cryo-EM and cryo-ET imaging techniques, you basically get the blob of density, but you don't always know what that protein was, right? It's not necessary that you know, like, this is this protein. And what you can do is you can run search against the AlphaFold database, which now includes structures of basically all proteins known on Earth, or maybe just, probably you know what organism that was, so you can restrict that a bit. And then you can identify the proteins in your experiment, which is a very big application for this kind of technology. Cool. So what's next? Are we done yet? I think this is just the beginning of this journey of AI in structural biology. The first thing we should do is understand proteins in the context of the cell. So in the cell, there is not just the single proteins or anything like that. There is all sorts of things. So as the first step, we have released AlphaFold Multimer, which can predict the structure of multiple chains. But there are also non-protein components like DNA, RNA, ligands, waters, ions, and all of them are important for understanding proteins. And on the right, you see RNA polymerase, which reads off the DNA and predicts and outputs the corresponding RNA. And another major challenges involve protein dynamics and effect of mutations on proteins. So proteins are not these bricks which don't move around. They're actually machines which look like this. So they wobble around. And this is, I think, SARS-CoV-2 spike protein. And we would like to understand this without the huge computational expense of molecular dynamics. So predicting the conformational states, the dynamics of proteins. And another thing which is extremely important, but is not covered by AlphaFold, is the effect of mutations of proteins. So when there is a mutation in the protein, like a single mutation, often the protein will, for example, stop working. Like if you make some mutations to the spike protein, it will break down. But the structure may not change. And that's the most interesting thing. Maybe the structure will stay the same, but the dynamics will change. And so there is this complementarity between understanding the effect of mutations and the protein dynamics. So hopefully we can make progress on that as well. Okay, any questions about this? Yeah. Okay. Okay, and now I would like to say thank you to the big and brilliant group of people who made all of this possible. And with that, thank you for your attention, and I'm happy to take any questions. Yes.
So, my question is, whether this supports sort of other interventions, like basically a change in environment? Because I always imagine that, like with my rudimentary biology knowledge, that folding highly depends on if the protein is in water or in oil or in acid.
Yes, I think this is one of the examples where currently it is sort of marginalizing out this experimental conditions. So most of the time people will do proteins in water, not in oil or acid, right? So basically the structures it has seen mostly correspond to the proteins in water. I don't think there is any reason why it can't be conditioned on that. The key reason it works like that is because of the CASP format, so where you basically only given the sequence and you predict the structure. So you can't assume you have access to this extra information. But I think it will be a big improvement to have that available as an optional intervention.
And also temperature would have an effect as well, right?
Yes, it does. Maybe a bit harder to incorporate, but yeah, we can talk offline. Yeah.
How long time was the project duration since the first idea to the results?
So I would say if you include AlphaFold 1, which participated in CASP13, probably until like this 5 years, probably, something like that. Yeah. So AlphaFold 1 to AlphaFold 2 is basically like, CASP to CASP, like 2-ish years. Yeah.
AlphaFold, can it predict function proteins?
No, it only predicts the structure, but I think you can modify it. There were definitely some works trying to use it for function prediction. Yeah.
I wonder, does AlphaFold help us understand the problem of protein folding? Because here we're resolving the structure, but the structure, if I understand correctly, is driven from the whole sequence as we have it, like the whole 5-year sequence of amino acids. And I was wondering, as the proteins are exiting the ribosome when they're created, the first amino acids are actually likely to start folding before the end of the protein is actually synthesized. So have you maybe tried to look at what happens sequentially as time goes on?
It's I don't think AlphaFold is the right way to study this. It's basically, as far as I understand, the big research question of how much this sort of process of folding actually affects the structure. And AlphaFold definitely just, again, it's one of the things it marginalizes over, because the main way you will get a protein is through this, like getting it one letter out of ribosome and then purifying and crystallizing. So it will have all of these biases already built in. And the problem is it's also trained on crops of proteins, which means that probably if you start adding one residue at a time, it will just try to guess what's next and sort of try to predict the structure for that. So yeah, AlphaFold, I think, is just not the right tool for this. Yeah.
So you didn't mention other ML methods to do protein structure prediction. I probably that attract a lot of attention in computation chemistry or computation biology. So I mean, and I guess they're based on energy minimization methods. So can you use your prediction as a starting point for this energy minimization problem and to have kind of an objective measure of how well your prediction is? So just the value of the energy, you see that.
So I think basically AlphaFold 1 was an energy minimization pipeline in the end. So it was basically produced like energy and then that was minimized. AlphaFold 2 removed that and it started working much better. I think the learning from that is that the fact that there is an energy function which you can minimize does not necessarily make the method more principled or better. The problem, unless you know that the energy is correct. But for proteins, you know that it's wrong, basically. So it's not quite as accurate as people thought, I think, which made it hard to get good predictions out with this way. And essentially, you just sidestep all this energy minimization thing and just let the network do it for you. Yeah. But I think there are some applications where it might be quite useful, and I think Rosetta community has been looking at that as far as I understand.
So writing down the energy function is actually hard. That's the main problem.
Well, yes, like you have to compromise. Like if you write it in terms of quantum effects, then you get like Schrodinger equation, and then it's accurate, but you can't solve it, and then you start making approximation, and rapidly becomes tractable, but not accurate. Yeah.
There is something I didn't quite understood at the start of the presentation. You spoke about evolution and the way mutation impacted. I didn't understand how you using this into AlphaFold in the model.
So we use this as the input. So let me go back to. So this multiple sequence alignment includes all the mutated versions of the sequence you would like to fold. So that's how it enters the model as an input.
Yes, and so if in your protein you find an alignment on your data, your portfolio of protein, then you use the 3D structure of this effective protein because you know it? Or
no, well, here it works fine with just the multiple sequence alignment, which only includes the sequences. So you only look at the sequences. But optional, you can also include the structures if you know the structures for some related proteins.
So what is the information that it brings you, the fact that in other proteins,
So this is basically this. So the most important one is probably the coevolution, where you know that if you see a correlation between columns of this matrix, then this means that there is some interaction between these two. So this, and the network is very good at parsing that.
Just so that they will potentially be close.
Exactly, yes. Not necessarily, there is a lot of spurious correlations. And it looks like the main thing AlphaFold is doing is precisely inferring which one are true correlations, which one are spurious. Cool. How much time do we have for questions?
I mean, you have a coffee break afterwards, so
oh, okay, fine, yeah. Nice.
We start a few minutes late.
Yeah, I have a couple of questions related to the small amount of labeled data you have. One is, did you clean up, I guess, from the database you were using, any low quality like protein structures?
Yes, I think there is some filtering, but it's not a very strong. Like, yeah, essentially, a PDB provides some level of, it's called resolution, so level of confidence in predictions, roughly speaking. And we also do some clustering. So another interesting thing about PDB is that people will upload whatever they get out of their results. So, for example, people use some proteins to calibrate their equipment. And there are like many, many thousands of structures for these proteins. But they're not very interesting to train on. You know, they shouldn't have much higher weight.
Yeah. One quick question is the distillation thing. When your ablation experiments, it seems removing the distillation made the model better. Distilling unsupervised.
Oh no. This is basically with self-distillation training. So the baseline did not use self-distillation. So this is the improvement you get. Yeah.
So in the team that work on AlphaFold, are there some protein specialists working with the artificial intelligence developer, or how did you get the knowledge about protein and this information about evolution?
So I think the team lead for AlphaFold is John Jumper, who basically did his PhD in proteins. So he had like a proper background in both ML and protein science. Most other people had knowledge of ML, biology, physics, chemistry, but maybe not the specific problem, but we learned on the go, I think. Yeah.
Yes, so I have a question about this whole evolution part. So I'm not sure I'm getting it. So aren't you just determining a correlation between the parts? Where is the evolution of the story?
Oh, you mean here?
Yes. Yeah.
Oh, the evolution comes from the fact that when you search the genetic databases, you will find evolutionary related sequences. So if you think of this, yeah, basically there is, you know, the tree of life which connects everything on Earth, and you essentially just want to find the same protein in evolutionary related organisms, right? So that you can align them, because otherwise you won't be able to align them, right? So this is how evolution comes in.
So you do this for all proteins, like for hemoglobin, and you have the structure of hemoglobin for cats, dogs.
You have the sequence for all of them, because the sequencing is very cheap. The structures you will have much less of. Yeah. Cool. Yeah, I'm happy to take more questions during the coffee break.
These are the name of the PDP1, PDP2.
I think so, yes.
And PDP1 is close to PDP2, then other.
Yeah, exactly, yes.
Okay, let's thank Michael again.